In this part, I want to talk about the histology of the urinary system. In this picture, you can see the overview of urinary system. As you see here, and as you know from our previous session, the urinary system is composed of two kidneys, and in this picture, you can see the left kidney. We have the indented part in the medial side of the kidney, which we call it hilum or hilus. And in each kidney, which is covered by fibrous capsule, you can see cortex around and medulla inside. The medulla is composed of darker renal pyramid and lighter renal column between the pyramids. The renal pyramids end with the renal papilla. The renal papilla open to minor calyx. Minor calyces merge together, make major calyx, and major calyces can make the renal pelvis and then ureter. And the ureter, as you see in this picture, open to the urinary bladder on the posterior wall, as you see in this picture. The ureter doesn't have any anatomic valve, and the detrusor muscle, which we have in the wall of bladder, can work as the sphincter for the ureter. And then the urinary bladder open to the duct, which we call it urethra, and you know that the structure of urethra in male and female are different from each other. The functional and structural unit of our kidneys are called nephron. And you know that the nephron are located both in cortex and medulla. Some parts of the nephron are in cortex and some parts are located in the medulla. And in this picture, you can see the structure of the nephron. The nephron is made up of glomerulus and the Bowman's capsule in the beginning. Glomerulus and Bowman's capsule together are called renal corpuscle. Then we have proximal convoluted tubule. Then we have loop of Henle, descending limb, loop shape, and ascending limb. And then we have distal convoluted tubule. Distal convoluted tubule merge to the collecting duct. The collecting duct can collect urine from up to 5 distal convoluted tubule. Then these collecting ducts merge together and make papillary ducts, and papillary ducts open to the minor calyx through the renal papilla. And you can see some blood vessels in this picture. You have the afferent arteriole, which is a branch of our interlobular artery. Then the afferent arteriole make glomerulus in the Bowman's capsule, and the efferent arteriole comes out. The efferent arteriole wrap around the nephron to make the peritubular capillary in the cortex and vasa recta in the medulla. And vasa recta and peritubular capillary, all of them merge together, make interlobular vein, and the interlobular vein goes to the arcuate vein. Each part of our nephron has the specific histology, has the specific microscopic structure. The proximal convoluted tubule has simple cuboidal epithelium with many microvilli, which we call them brush border. The distal convoluted tubule is composed of simple cuboidal epithelium, and we do have the microvilli here, but the number of our microvilli are very few. You can see different parts of loop of Henle here. The thin parts, the thin segments composed of simple squamous epithelium, while the thick part is simple cuboidal. And you can see capillaries with simple squamous endothelium between these ducts. Our papillary duct and collecting duct has simple cuboidal epithelium, and the interesting thing about these two ducts are the lateral borders which we have between the cells are very visible, and I will show it to you in the next slides. You have one slide in your PowerPoint. It talks about all parts of the cortex and all parts of the medulla. Just, I want to go back to the previous picture and show you which parts of the nephron are located in the cortex and which parts are in the medulla. I draw a line here to show you the cortex, which is located here, and the medulla, which is here. As you see in this picture, the renal corpuscle, proximal and distal convoluted tubule are located in the cortex, while the loop of Henle, descending and ascending and U-shaped part, the last part of the collecting duct and the papillary duct are located in the medulla, and the blood vessels are everywhere. So when you want to check the histology structure of the kidney, you need to pay attention which parts you can see in the cortex and which parts are located in the medulla. Now, in this picture, you can see kidney histology. And in the kidney histology, you can see cortex and medulla. I magnify it a little bit to show you what do we have in cortex and what do we have in the medulla. I draw a line between cortex and medulla here. This blue line represents the boundary between cortex and medulla. When you look at the cortex, you see that we have some roundish structures, which we don't have it in medulla. So one of the important differences between cortex and medulla are these roundish structures. They are renal corpuscle, which is composed of glomerulus and Bowman's capsule, and I will show it to you in the next slide. The next part, the next differences which we have between cortex and medulla is the color of cortex and medulla. The cortex is deeper in color, while the medulla is lighter and pale. We have proximal and distal convoluted tubule. Besides the renal corpuscle in the cortex and in the medulla, we have mostly the collecting duct, the papillary duct, and the loop of Henle. And as I told you, the blood vessels are located everywhere. Now, I go to higher magnification and I show you the cortex and medulla. In this picture, you can see cortex and medulla, and the differences between these two parts are very, very visible. Again, the blue line here can show you the boundary between cortex and medulla. In this side, we have cortex. In the other side, we have medulla. In the cortex, you can see the renal corpuscle. As I told you before, the renal corpuscle is composed of glomerulus and Bowman's capsule. So you can see glomerulus here, and the Bowman's capsule is located around. The Bowman's capsule has two layers. One layer is visceral, which is attached to the glomerulus, and one layer is parietal, which is located outside. Between visceral and parietal, you have capsular space where the filtrate can flow here. Then you can see the proximal and distal convoluted tubule. I want to tell you the differences between them. The proximal and distal convoluted tubule, both of them has simple cuboidal epithelium. The difference between these two is this. We have more reabsorption in proximal convoluted tubule. So this tubule needs to have more mitochondria in the epithelial cells. Due to the abundance of mitochondria here, the color of our cells become deeper when we compare it with the distal convoluted tubule. The other differences is the microvilli, which we have in proximal, and we don't have it that much in distal. So the lumen of the proximal is smaller, while the lumen of the distal is larger. I show you more pictures, and we can see these differences better. And this is the medulla of the kidney. In the medulla of the kidney, you have two types of ducts. One type of duct are collecting and papillary. They are pale in the color. The other one is deeper in the color, and they are different parts of loop of Henle. And as I told you, the interesting thing about the collecting ducts are the lines which you can see and very visible between the cells. These lines are called lateral border of the cells, and we can see it in collecting and papillary ducts very, very clear. Here you can see the cortex of the kidney. In the cortex, as you see in this picture, we have renal corpuscle. So when you see renal corpuscle, don't doubt about you are in the cortex of the kidney. And then you have two different types of ducts around. One duct is proximal. You see that the color is deeper. And in the lumen, you have some fuzzy structures which come inside. These fuzzy structures are microvilli, which we have here. They protrude to the lumen, and they make our lumen become smaller. While in distal convoluted tubule, you see that the color of the cytoplasm of the epithelial cells are pale, and the lumen is bigger. Again, here you have distal convoluted tubule, and the others are proximal. And we have a section of collecting tubule here with the lateral borders, which are very visible between the cells. For the renal corpuscle, you have glomerulus inside, and the visceral layer of the Bowman's capsule is attached to the glomerulus, and the parietal layer is outside. Between visceral and parietal, you have this space, which we call it glomerular space or capsular space. And here is the medulla. As you see in this picture, in the medulla, we don't have any renal corpuscle. You have some darker ducts. They are very deep in pink color. They are different parts of loop of Henle. Some of them are lined with simple cuboidal epithelium. They are thick segment. Some of them are lined with simple squamous epithelium. They are thin segments. You can see many blood vessels between them. You have RBC in these blood vessels, and you have some other tubes with a very distinct lateral border between the cells. Look at here, the lateral border is these borders, which you can see between the cells. And it's only visible in the collecting and papillary duct. This is a duct. And these lines, which you see between two adjacent cells, are lateral borders. And as you see here, these lateral borders are very visible in collecting and papillary duct. This is collecting, and this is the papillary duct. And all of them shows you these lateral borders. The next structure which I want to show you is the structure of the ureter. The first thing, the first key for identification of the ureter is the star-shaped tube or lumen which you have. Look at here, the lumen is very similar to the star. And this is a key for identification of the ureter. Your epithelium is transitional epithelium. You know that in transitional epithelium, we have multiple layers and the surface cells are pillow-shaped. I show you higher magnification of the transitional later. Then you can see lamina propria and tunica submucosa, which are mixed and intermingled together because the muscularis mucosa is absent in the ureter. And then you have tunica muscularis externa with inner longitudinal and outer circular layer. The orientation of the muscles in the urinary system is opposite to the digestive system. And the last layer is tunica adventitia with many adipose tissue. This is higher magnification of the ureter. In this magnification, you can see the transitional epithelium here. You can see the lamina propria and tunica submucosa. These are connective tissue which mix together. You have longitudinal and circular smooth muscles after that. And then you can see the tunica adventitia. In this picture, you can see the urinary bladder. In the urinary bladder, the first thing which you need to pay attention is the folds, many folds which you have here. The folds which is made by epithelium and lamina propria. We call them rugae, and you have rugae in the urinary bladder. Your epithelium is transitional. This is the lamina propria, and here you have tunica submucosa. We have a very discontinuous muscularis mucosa here. Then you can see thick layers of muscles. The inner and outer layer are longitudinal, and the middle layer is circular. The muscles together, all of these layers are called detrosor muscle. And after that, you have tunica serosa, connective tissue with the coverage of mesothelium outside. And this is the other picture of the urinary bladder. In this picture, I want to show you the transitional epithelium when they are relaxed and when they are in a stretch. When they are relaxed, they are pillow shape or umbrella shape. But when they become a stretch, the surface cells become flat. This is the characteristic of transitional epithelium, to become flat when the urinary bladder is full of urine and provide larger space for storage of urine. You can see the lamina propria after the epithelium, a very discontinuous muscularis mucosa, tunica submucosa, and after that we have muscle layer. The muscle is detrosor muscle. And if you remember from my lecture last time, I told you that the transitional epithelium can show and can contain some binucleated cells, the cells which contain two nuclei, like this one or this one, or the other one is here. Binucleated cells. We don't know why do we have binucleated cells here. This is only the structure of these cells. Now you can see the slides, the real histology pictures of these different parts. Here you have the cortex of the kidney in this picture, and you have a large renal corpuscle. Your renal corpuscle is composed of glomerulus, which is encircled in yellow, and we have capsular space and the parietal layer of the Bowman's capsule here. And you have two types of ducts here. This duct is proximal and this one is distal. The difference between them is, first, in the proximal, the epithelium is very pink, while in distal, it's pale pink. In the proximal, you have fuzzy structures which come to your lumen. They are microvilli, but in distal, you don't have them. This is the other proximal convoluted tubule. This is the other view of the renal cortex. You can see the renal corpuscle here, the glomerulus, Bowman's capsule. This is the space of the capsule. Here is parietal layer, and attached to the glomerulus, you have visceral layer. And around the glomerulus and Bowman's capsule, you can see some proximal convoluted tubule. You see that all of these tubules, which you can see here, are pink, very pink in their color, and their fuzzy microvilli come to the lumen. The other picture, this picture, I want to show you the proximal and distal. Both of them have simple cuboidal epithelium, but in the proximal, you have very pink epithelium. In distal, epithelium is pale, and you can see fuzzy pinkness inside the lumen in proximal, while we don't have it in distal. And you know that in the proximal convoluted tubule, we have many reabsorptions. Due to that reabsorption, we have microvilli here. This is the other picture of the renal cortex. In this picture, you can see glomerulus, Bowman's capsule around, and you have one proximal convoluted tubule which immediately come out of the Bowman's capsule. And these are the proximal two by all of the informations which I gave you before. And this one is distal. You see that the color is different from proximal, and we don't have that microvilli in the lumen. The other renal cortex, pay attention to the glomerulus and Bowman's capsule and the ducts which we have here. Number two, like this one, this, or this one, are proximal convoluted tubule. You see deep pink and fuzzy appearance in the lumen. But this duct is distal, or this one. These two are distal because the color of the epithelium is pale and the lumen is wider, and you don't have that much fuzzy microvilli inside. And this is the collecting duct because you have lateral borders between the cells. But you don't need to know about the collecting ducts in this level. In this picture, I want to show you the microvasculature in the kidney. You know that in the cortex of the kidney, we have some branches of interlobular artery. Interlobular artery make a branch which we call it afferent arteriole. Afferent arteriole make glomerulus. And in this magnification, I can't show you the efferent, but you can see the peritubular capillaries, all of these blood vessels which wrap around the tubes, the different parts of the nephron in the cortex. In the next picture, you can see the interlobular artery. You have afferent arteriole, glomerulus, and efferent arteriole. The efferent arteriole can make peritubular capillaries. Afferent, glomerulus. This is the picture of urinary bladder. histology picture. In this part, you have the lumen of the bladder, then you have tunica mucosa. Here you have tunica muscularis and tunica serosa. Now I go to higher magnification. In tunica mucosa, you can see transitional epithelium and lamina propria. And in tunica muscularis, you can see inner and outer longitudinal and middle circular. Together, all of this layer is called detrosor muscle. Now, let me show you, this is inner longitudinal and this is outer. And between them, you can see the middle circular. These are the layers which we have in the wall of urinary bladder. Detrosor muscle, all of them together. This is the transitional epithelium. Look at the arch shape or umbrella shaped cells in the apical layer of this epithelium, which become stretched and provide larger space. Lamina propria, and this is the muscularis layer, which is the detrosor. The other urinary bladder, you can see transitional epithelium and lamina propria, discontinuous muscularis mucosa, then you have tunica submucosa and detrosor muscle. And this is the zoomed transitional epithelium. When you look at this epithelium, you have multiple layers and the surface cells are umbrella shape. You can see binucleated cells here. And this is the picture of ureter. I told you that in the ureter, the best key for identification is the star-shaped lumen. Your epithelium is transitional, lamina propria, then you have tunica muscularis externa, and then you have tunica adventitia with many adipose cells. And this is the last slide of today. In this slide, you can see ureter again, the star-shaped lumen, the transitional epithelium, lamina propria, and then you have muscularis externa, and the last layer is adventitia.